Have you ever wondered how a country with some of the most challenging, extreme terrain on Earth is literally hacking plant biology to climate-proof its entire food supply? Well, welcome to this explainer. Today, we're taking a really deep dive into Nepal's incredible journey into plant biotechnology. We're going to see exactly how scientists are mapping genetic blueprints and engineering crops to not just survive, but actually thrive in a rapidly changing world. So here's our roadmap for this explainer. We'll quickly define the biotech foundation, check out Nepal's institutional ecosystem, unpack their precision toolkit, look at mapping the genetic blueprint, see how they're transforming the crop portfolio, and finally, look at engineering a sustainable future. Let's get right into it.
Section 1. Defining the Biotech Foundation. Plant versus agricultural biotechnology.
All right, let's get our definitions straight first. On one side, we have plant biotechnology. You can think of this as the big, comprehensive toolkit. It's the broad science, the general screening and foundational genetic manipulation used to adapt plants for specific needs. But then, on the other side, we have agricultural biotechnology. And this is where things get laser-focused. It's a highly applied science. Instead of broad strokes, agricultural biotech is all about making incredibly precise genetic changes. We're talking about taking highly beneficial, specific traits, things like pest resistance, disease resistance, or herbicide tolerance, and placing them directly into the crops farmers are actually growing out in the field. So plant biotech is the broad foundation, right? While agricultural biotech is the pinpoint application.
Section 2. Nepal's institutional biotech ecosystem. 30 years of progress.
You know what's really amazing here is that Nepal didn't just wake up yesterday and start doing this. They've been building a dedicated ecosystem for this advanced science over the last 3 decades. Back in the early 1990s, the National Agricultural Research Council, or NARC, started integrating basic biotech tools to just boost the efficiency of their breeding programs across different stations. Then, jump to 1998, they established the Kumultar Hub, which is the National Biotechnology Research Center. At first, they focused on micropropagation and tissue culture for foundational crops like rice, wheat, and sugarcane. And that brings us to the present era, where it's all about institutional synergy. Today, the Center for Biotechnology at the Agriculture and Forestry University, AFU, is collaborating directly with the Department of Genetics and Plant Breeding, and they are heavily focused on advanced molecular marker technology. It's been a massive evolution.
Section 3. The Precision Biotech Toolkit. Diagnostics and Propagation.
So, if we look at the toolkit driving all this, it really breaks down into 2 distinct pillars. First, there's the diagnostic pillar. That's molecular marker technology. This is basically reading the manual. It's all about mapping the DNA, assessing diversity, tagging genes, and building linkage maps so scientists know exactly what's hiding inside the plant's genetics. Then, you have the propagation pillar, which is tissue culture. And this side, it's all about completely bypassing traditional biological timelines, using techniques like micropropagation and culture. So one side reads the genetic code, and the other side physically accelerates how the plant grows and reproduces.
Let's zoom in on that tissue culture pillar for a second, because honestly, these methods are essentially biological cheat codes for scientists. First up is culture. I like to think of this as the time shortcut. By taking advantage of highly responsive stages of tissue culture, especially in crops like wheat and rice, scientists can actually produce double haploid lines. And the payoff? It drastically reduces the breeding cycles. What used to take years and years of waiting can now be completely fast-tracked. Then there's embryo culture, which is the biological shortcut. Look, sometimes when you try to cross 2 different plant species, nature just puts up a wall, a reproductive block. In vitro embryo rescue entirely bypasses those natural barriers, letting scientists successfully produce interspecific and intergeneric hybrids. It's basically rewriting the rules of what's possible.
Section 4. Mapping the genetic blueprint. The path to precision.
Getting to that level of precision doesn't happen by accident. It requires a very specific sequential path. Picture this scientific pipeline like a giant funnel. First, you start super wide with diversity assessing. You have to establish the baseline genetic variation in both wild and cultivated species, basically figuring out what you're working with. Once you know what's out there, step 2 is creating linkage maps and gene tagging. This is where you identify exactly where specific traits live on the genome. Step 3 narrows it down even further with QTL mapping. That stands for quantitative trait loci, which uses DNA markers to locate those really complex traits. And finally, at the very narrow tip of the funnel, you arrive at marker-assisted selection, where you can actually start stacking these beneficial traits together. And that brings us to a concept that is absolutely crucial to understand: marker-assisted selection, or MAS. This is the holy grail, the ultimate goal of all that foundational mapping work we just talked about. It allows breeders to use DNA markers to stack beneficial genetic traits with pinpoint precision. It literally takes all the guesswork out of traditional plant breeding.
When you look at this process as a whole, starting wide with diversity assessing and moving step by step all the way down into the specifics of marker-assisted selection, you realize something really important. These preliminary mapping works are an absolutely mandatory foundation. Without going through this step-by-step funnel, advanced precision selection is simply impossible. I mean, you cannot select what you haven't first mapped, right?
Section 5. Transforming Nepal's crop portfolio, from labs to fields.
Now, Nepal isn't just trying this out on 1 or 2 test plants. They're deploying these advanced diagnostics across a massive, super diverse portfolio. We're talking about using specific diagnostic markers like isoenzymes and RAPD primers across the board. They are monitoring genetic variations in everything from staple cash crops like tithe and wild buckwheat to fruits like citrus and mango, all the way to pigeon pea and taro. They're even using specific isoenzymes to assess the genetic diversity of Swerchia, which is a highly vital botanical plant. The sheer scope of this genetic cataloging is honestly incredible.
But things get really interesting when we look at major cereal and staple crops, because that's where the approach gets incredibly tailored. Take barley, for instance. They're doing broad spectrum mapping there, deploying over 90 specific SSR markers and multiple isozymes just to estimate the diverse population parameters. But then, if you look at wheat, it shifts to highly targeted trait diagnostics. They use allele-specific diagnostic markers to pinpoint crucial real-world traits. For example, they've mapped out reduced height genes, specifically RHT-B1b and RHD1b. Now, why does that matter? Because those genes prevent the wheat from growing too tall, just falling over in the wind. They're also identifying photoperiod genes, which literally control when the plant decides to flower. We are looking at precision engineering applied directly to a nation's staple food.
Section 6. Engineering a Sustainable Future. The Next Generation. All of that intense DNA mapping and tissue culture we've talked about, it culminates in the next generation of crops. Take a look at these 2 wheat varieties currently in the release process. On one hand, we have AFU01 wheat. Its core superpower is high nutritional value. It has a high protein profile, making it absolutely perfect for premium bread and chapati production. But then compare that to AFU02 wheat. This variety is all about extreme climate resilience. It's engineered specifically for hardcore heat and drought resistance, and it's heavily optimized for late-sown conditions. It really grounds all that complex lab science into vital, real-world outcomes. You get superior nutrition over here and tough-as-nails climate survival over there. But, and this is a massive but, advanced plant biotechnology absolutely has to be paired with sustainable agronomic practices if you want to realize its full yield potential. Think about it. You could have the most genetically perfect wheat seed on planet Earth, but if your soil is dead, that crop is going to fail. Period. And that's exactly why Nepal is integrating this high-tech genetics with soil health through efforts like the Biochar initiative. This push for biochar utilization is specifically designed for both immediate soil health improvement and long-term, sustainable crop yield preservation. The genetics and the soil simply have to work together in a continuous, sustainable loop.
So, if we want to summarize this entire journey, we can look at it as a clear 3-step blueprint. First, Nepal is cataloging the baseline. They are using decades of rigorous diversity mapping to essentially fingerprint their unique land races and wild varieties. Second, they're engineering precision. They're using that toolkit of tissue culture and allele-specific diagnostics to completely bypass biological barriers and isolate vital traits, like those reduced height genes we talked about. And finally, this all leads to securing the future, which culminates in the direct release of highly authenticated, climate-resilient, and nutritionally superior crops, just like those AFU wheat varieties. By transforming foundational genetic mapping into targeted marker-assisted selection, Nepal is actively and successfully engineering its agricultural future. Which leaves us with a pretty massive final thought. As climate change accelerates and threatens food supplies all over the world, could Nepal's unique approach, pairing ultra-precise genetic mapping with ancient foundational soil health practices like biochar, could that become the definitive global blueprint for survival? It's a fascinating question for sure, and definitely one we'll be watching closely. Thank you so much for joining me for this explainer,